Hello students, in the previous tutorial we studied a Mendelian inheritance pattern that was dominance, example of which was that of pea, wherein we crossed a tall pea plant with a dwarf pea plant, and we noticed that the F1 hybrids were all tall, resembling one of the two parents. The second case that we studied was of incomplete dominance, and that is a non Mendelian pattern of inheritance because here Mendel's law of dominance is not followed. The example we saw here was that of Snapdragon, wherein when you cross a red variety with a white variety, red being dominant over white, we saw that in the F1 hybrids, the phenotype that was expressed was pink, indicating that red was not completely dominant over the white gene. Okay, so we have studied two patterns of inheritance and dominance so far. One was complete dominance, that is the Mendelian pattern of inheritance, and a non Mendelian pattern of inheritance, that is incomplete dominance. I hope you were clear with that. Today we are studying another non-Mendelian inheritance pattern that is called as codominance. So what is codominance? In codominance you will see that both the alleles that are there in the gene pair are dominant and express fully. Therefore we say it is codominance. That means both the partners are expressing their dominance together. An example of it in human beings is in blood groups. So I think you are all aware that there are different types of blood groups in humans, and these blood types arise because of the alleles of a gene called as I. So the I gene has different alleles, and to be specific it has three different types of alleles which control the phenotype, which is your blood type. So your blood type could be a blood group or a B blood group or a O blood group, right? So they are under the control of the gene I. Now this gene I has, as you can see, three different types of alleles. So you have the allele IA, which is responsible for producing the A antigen, IB, which is responsible for producing the B antigen, and I, which is not going to produce any type of antigen. So, what exactly is this antigen and where is it found? So, a person who has, say, the IA allele in his or her gamete, okay, in his or her blood cell, that is your red blood cell, will produce an antigen on the surface called as the A antigen. Okay, if the person, however, has this particular allele of the gene I, that is the IB allele of the gene I, then that person's blood red blood cell will be having, the surface will be having an antigen B. But people who have the recessive allele small I will not produce any antigen. Now let us see how this becomes an example of codominance. Okay, so this sentence is very important. If a person, we know that people are diploid organisms, right? So their gametes will have two different alleles of the gene. You can never have three alleles of a gene. You will have one allele coming from the father and one allele coming from the mother. Therefore they are diploid and they have two alleles of a gene at a time. So you have, suppose a person is receiving an IA allele from the mother and an IB allele from the father. So the person has a combination of IA, IB. What will happen in this case is the person will have the blood type of AB because both these alleles are dominant alleles, thereby they are written in capitals, and both are expressing. Okay, so this is an example of co-dominance, because here it is that both are dominant. There's no allele that is recessive and hidden. But suppose the combination, you have received say IA from the father and small I from the mother, and then the person's combination is IA small I. Okay, so in that case, this is a dominant allele and this is a recessive allele, right? What is the blood group of the person? The person's blood group will be A, because IA is a dominant allele. Similarly, if the person is receiving IB from one parent and I from another parent, what will be the blood group of the person? B, because IB here is dominant over I. Okay, so if you've understood this blood group and genotype chart will be quite clear. If the person has a group, now blood group is the phenotype or the expression. That means if I am A blood group type of a person, that is my phenotype. What will be my genotype? So my genotype will either be homozygous, wherein I have both dominant A alleles, or it could be heterozygous, wherein I have received a dominant from one of my parent and a recessive from one of my parent. Similarly, if a person is B blood group, then the genotype possibilities are IB, that is homozygous dominant, or heterozygous dominant. If a person is AB, it's an example of codominance, you can see, wherein both the alleles are dominant and expressing, and therefore we have both the antigens, A antigen and B antigen, and that is your A blood group phenotype. O is if you have both recessive alleles. So this is homozygous recessive. Okay, so if a person is homozygous recessive, then and only then will the blood group of the person be O, because you do not have any of the antigens produced, that is A or B. So we move on to our next chart, which shows us the possible phenotypes of offspring if a cross is made between two parents who are A. Okay, so let us consider these cases. So if the father and the mother have a blood group of A, what will be the possibilities of the offsprings? Now, we have three cases here, or three scenarios. If the father and the mother are homozygous dominant A, the possibility of phenotype of the children will be just A. So all children born to this couple will definitely be homozygous dominant A. If the parents are one homozygous dominant and one heterozygous dominant A, then the possibilities of the children again could be that they could be homozygous dominant A's or heterozygous dominant A's, because suppose you get a IA from one parent and small I from one parent, then you become heterozygous A. But if you receive both dominant alleles from the father and the mother, you become homozygous dominant A. Now, suppose both parents are heterozygous dominant, there is a chance of you getting an O blood group. How? Because if you have small I from here and small I combining from here, you will have a O blood group. Otherwise, this A could be a homozygous dominant A, that is capital IA, or it could be a heterozygous dominant A. How? IA and small I. Okay, so if IA comes from here, small I comes from here, it is a heterozygous dominant A. So these are your possibilities. Now suppose the parents crossed up B. You have similar outcomes just like this. What I explained to you, just replace the A with the B. You will have, in this case, homozygous dominant B individuals. Here you can have homozygous and heterozygous individuals. Possibilities are probable. Here the probabilities are that you have a homozygous dominant B, a heterozygous dominant B, and a O possibility is there. Okay, then finally AB. Now consider this. So suppose you have a AB individual married to another AB individual, father and mother both AB, Please note, you can get, suppose you get a IA from here and IB from here, the person will be a, the offspring will be AB. But suppose you get a IA from here and an IA from here, you get a homozygous dominant A individual. If you get a IB from here and an IB from here, you get a B. So three probabilities are there. The offspring could either have a blood group of AB, A or B. And finally, O. Now, suppose if two individuals who are O, both father and mother are O, there is no chance of a child being any other blood group other than O, because you can see here there is only one possibility of combination, I from here and a small I from here. So based on this chart, you can get problems. Okay, asking you to guess what is, say, the genotype of the father given the genotypes and the phenotypes of the offspring, or it could be vice versa, wherein you are given the the genotype and phenotype of the father and mother, and you are asked to predict the genotype phenotype of the children. So we will come across, we will do those problems a little later. Okay, now coming to a conclusion of this part, what are the things to remember is that ABO blood grouping is not only an example of codominance, but it is also an example of multiple allelism, because as you can see here, the gene I is not just having two alleles, it has three alleles. Okay, so it's a example of multiple allelism also. And these alleles govern the same character of blood group. Now, when we study an individual, we don't have multiple allelism coming into play, because an individual is diploid and a diploid individual has only two alleles at a time, right? So, but when you take up population studies, you will see the effects of all these three alleles, A, IB, and I. Now, the next thing that's mentioned in your book is also important, and that is an example in pleiotropy. Now, the word is not mentioned here, but I will be putting the definition and the term in the chat box. Okay, so whatever important points are there will be listed out in the chat box. Please copy and paste it, or you can write it down in a in your book. You can copy and paste in a separate document on your system, or you can write these points in your text, but please keep note of these important points. So the final thing in this tutorial is about Pleiotropy. Now, what is pleiotropy? The definition is very simple. It is about one gene or one gene product which produces more than one effect. So you consider a situation like this where one gene is controlling more than one effects. So example in humans also are there, but they're not covered in your syllabus. The example in plant is given in your syllabus, and that is of starch synthesis in pea seeds. Now, if you see, starch synthesis in pea seeds is controlled by a gene called as B, which has two forms or two alleles, capital B and small b. But this gene B is not only controlling starch synthesis, that is one phenotypic effect, but it is also causing another phenotypic effect in the seeds, that is of their seed shape, that is either round or wrinkled. So you can see the same gene having two effects. One is of how much the of starch is synthesized, the starch synthesis part of it, and it is also controlling the shape of the pea seed. So you can see here that if a person is if a plant is a homozygote, that is capital B, the starch synthesis very effective, larger starch grains are produced, and in such seeds the shape is found to be round. Whereas if the person is if the plant is a homozygote which is recessive, capital small b, there's lesser efficiency in starch synthesis, and the seeds are found to be wrinkled. So you can see here two effects being controlled by the same gene. Heterozygote, capital B, small b, the seeds are also round, but they have intermediate size and intermediate starch synthesis. Okay, so this is an example of pleiotropy, as I told you, a single gene or a single gene product controlling many effects. Okay, with this we'll stop at this. What we have done in this part is we have studied another non-Mendelian inheritance pattern, codominance, one in blood groups. Okay, so it is also an example I told you of multiple alleles. And second, we have seen pleiotropy, which is again non-Mendelian inheritance pattern, wherein one gene is controlling many effects. Okay, and that is seen in pea plants. Any questions? Please keep your questions ready and we can solve it in our next session. Thank you.